If you work in a clinical lab, you see Klebsiella pneumonia on reports pretty much every single day. But what if I told you that a good chunk of those reports are wrong? Today, we're pulling back the curtain on a pathogen that's been hiding in plain sight for decades, Klebsiella variolica. It's a really important member of the Klebsiella pneumonia species complex, and its story is all about mistaken identity, surprising virulence, and a really unique connection to the world well beyond the hospital.
And this question, right here, gets to the absolute heart of the issue. What happens when that routine Klebsiella pneumonia you report out is actually masking a completely different and potentially more dangerous organism? This isn't just a what-if scenario. It's a reality playing out every day in labs that haven't updated their diagnostic methods. And, it has serious, real-world consequences for our patients.
Just how big is this problem? Well, it's pretty startling. Some studies that have gone back and reanalyzed old isolates with molecular methods found that up to 11% of bugs originally called Klebsiella pneumoniae were actually Klebsiella variicola. Think about that. It means we've been systematically underestimating its true role in human disease for a very, very long time.
Okay, so here's the game plan for this explainer. We're going to start with its identity crisis, then we'll dig into its clinical impact and what makes it so virulent. After that, we'll get into the nitty-gritty of lab identification, super important, cover treatment and susceptibility, and then wrap things up by looking at infection control from a much bigger One Health perspective.
All right, first up, let's unpack this whole identity crisis. Because to really get a handle on Klebsiella variolica, you have to understand where it comes from, taxonomically speaking, and why it's been so good at flying under the radar.
So, the root of the problem is right here. It's what we call the Klebsiella pneumoniae species complex, or KpSC for short. These organisms are basically siblings, so closely related that on the bench, using our classic biochemical tests, they look practically identical. For years, Klebsiella variolica has been the ghost in this machine.
The journey to actually recognize this bug was a slow one. It really started back in 2001 when sequencing work identified this distinct group called Kp3. Then, in 2004, it was made official. Rosenblatt and his team formally named Klebsiella variolica. And what was so cool is they found it in both human patients and in plants, like banana roots. And that brings us to today, where, even though we know it exists, actually identifying it correctly is still a huge challenge.
So why does all this matter so much? I mean, who cares about a name, right? Well, it matters because getting the ID right isn't just an academic exercise for us microbiologists. It is absolutely critical for patient outcomes.
And that's because, clinically, Klebsiella variolica is a major league player. This is not some minor, insignificant bug. It's behind a whole spectrum of really serious infections. We're talking bloodstream infections, UTIs, pneumonia. And it's especially worrying as a cause of neonatal sepsis, where we've seen outbreaks in NICUs that have had devastating consequences.
Now this brings us to something that's both fascinating and really concerning, what some are calling the mortality paradox. Just look at this data from a big study out of Stockholm. For bloodstream infections, the 30-day mortality rate for Klebsiella variolica was nearly 30%. That is more than double the rate for Klebsiella pneumoniae in the exact same study. It's a pretty shocking statistic, right? Especially when other studies suggest it actually has fewer of the classic hypervirulence genes.
So what gives it this edge? What makes it so pathogenic? Well, it's got this standard Klebsiella toolkit, that protective capsule, systems for stealing iron from our bodies. But when scientists dug into its genome, they found something really unique. It has a P. pilus operon, a well-known weapon for uropathogenic E. coli, but something never seen before in the Klebsiella genus. This could be the secret to its success in UTIs and maybe even that crazy mortality rate.
Okay, this all leads us to the most critical part for those of us on the bench. How do we actually find this thing? It's time to move past the old, ambiguous biochemical tests and get into the certainty of modern methods.
So if you're looking at a plate, it's a classic Klebsiella, gram-negative rod, of course. And on a McConkey plate, you're going to see those big, pink, often goopy-looking colonies from the lactose fermentation. You've seen it 1,000 times. And at this stage, it's completely indistinguishable from its more famous cousin.
And this right here is the money slide. It's all about choosing the right tool for the job. Our automated systems like the Variicola 2, they're just not going to cut it for telling these species apart reliably. Now, MALDI-TOF, that's a game changer for speed, But, and this is a huge but, its accuracy is 100% dependent on its database. If your lab's library hasn't been updated to include the specific protein fingerprints for Klebsiella variicola, we're talking Bruker version 9 or higher, it will keep calling it pneumoniae. For confirmation, a multiplex PCR that targets a specific gene like YGGE is fantastic. And of course, sequencing a gene like RPOB is still our ultimate gold standard.
So once we have that accurate ID, the next big question is, what do we do about it? How do we treat it? Let's take a look at its built-in resistance profile and what the current guidelines are telling us.
First things first, and this is non-negotiable. Every single isolate of Klebsiella variicola carries a gene for a beta-lactamase right on its chromosome. What that means for us is built-in intrinsic resistance to penicillins like ampicillin and amoxicillin. These drugs are useless against it and should always be reported as resistant no matter what the in vitro pest might look like.
Now, for empiric therapy, that first guess before we have all the data, we can look to solid guidelines like these from the Dutch SWAB group. For syndromes where you suspect an Enterobacterales, like urosepsis or a hospital-acquired sepsis, a third-gen cephalosporin or pip/tazo are your go-to starting points. But the key, as always, is to de-escalate to a more targeted drug once you get those final ID and susceptibility results back from the lab.
For the really tough cases, the multi-drug resistant strains, the 2024 guidelines from the IDSA are essential reading. For ESBL producers causing infections outside the urinary tract, carbapenems are still king. But when you get to carbapenem-resistant isolates, the recommendations get really specific, depending on the type of carbapenemase and where the infection is, often using these newer combos like ceftazidime-avibactam, or even a combination of two drugs for those tricky MBL producers.
And of course, none of the therapy guidelines matter if we can't interpret our susceptibility results correctly. This is where standardized breakpoints from groups like EUCAS are so incredibly important. These standards are the rulebook. They are the crucial link that lets us take a raw MIC number and turn it into something clinically actionable, susceptible, intermediate, or resistant.
Okay, for our final section, let's zoom out. We're going to broaden our perspective beyond just treating the patient in front of us and look at infection prevention. And for Klebsiella variolica, that means looking way beyond the hospital walls. And this slide right here perfectly illustrates why this bug is such a One Health challenge. On one side, you've got your usual suspects in the hospital: hands, sinks, medical gear. But then look at the other side. Klebsiella variicola is thriving out in the environment. It's on plant roots, it's used as a commercial biofertilizer, it's even been pulled out of city rivers, sometimes already carrying carbapenemase genes. It's basically living a double life.
Now, back inside the hospital, the playbook for any multi-drug-resistant bug is pretty clear and backed by a ton of evidence. It all starts with hand hygiene. It's the single most important thing. Then you add contact precautions, active surveillance for high-risk patients, really good environmental cleaning, cohorting patients during an outbreak, and of course, strong antimicrobial stewardship to take the pressure off.
I think this quote just nails the ultimate challenge here. The fact that Klebsiella variolica can so easily jump between a hospital, a farm, and a river means that a resistance gene that pops up in one place can easily find its way to another. A resistance gene from a hospital drain could, theoretically, end up in our food supply. That's a pretty sobering thought.
So, what's the big takeaway from all this? Well, to really fight back against Klebsiella variicola, we need a three-pronged attack. It has to start in our labs, with accurate and modern diagnostics. It has to continue on the hospital wards with relentless infection prevention. And it has to extend out into the community with a truly collaborative One Health approach to surveillance and stewardship.
And that wraps up our deep dive into Klebsiella variicola. Just so you know, all the information we talked about today was sourced using Gemini Deep Search, and I really encourage you to go check out the full list of citations in the linked notebook document. If you found this explainer useful, please hit that subscribe button for more breakdowns of important topics in clinical micro. We'd also love to hear your thoughts or questions, so drop them in the comments below. Thanks so much for joining us.